Hello and welcome everyone, and welcome to today's webinar. Today we will be discussing applications of demethylation across diverse fields. So let's get started. So first we'll begin with the biological background and epigenetics and the demethylation technologies. Then we will walk through the most commonly used demethylation detection technologies and discuss their principles and applications. And after then, we will explore how demethylation is being applied in cancer research and the development of disease biomarkers. And finally, we'll explore how recent advantages in plant research and multi-omics integration in demethylation, demonstrating how demethylation can be used to unravel complex biological mechanisms. So first of all, before we dive into applications cases in different research files, let's first take a look at the biological background of demethylation. And so for decades, DNA has been considered the molecule that stores genetic information in living organisms. But the new study in recent years shows that the genetic information is not fully decided by genes. The researchers have been found that we can change the modifications of genome without affecting the DNA sequence, and this change can not only influence an individual's development, but also can pass on the next generation. And there are two biological phenomena shown here. The first one is identical twins. They share exactly the same genome, but they often show differences in personality, in lifestyle, and other traits. And the second case is about queen bees and worker bees. They originate from genetically identical larvae, but they develop into very different individuals because of differences in nutrition during their development. And so it's a question that what is epigenetics? In 1942, the concept of epigenetics was first proposed to describe changes in that occur without change in genotype. And a wide variety of the epigenetic modifications have been discovered so far. And they have different modifications at different molecular levels, such as the DNA levels, the RNA levels, and the chromatin organization, and also on the histone. Okay. So among the many types of epigenomics modifications, demethylation is one of the most studied, which means a methyl group to the 5th carbon of cytosine in DNA sequence. And the studies have shown that the mammalian genome contains CpG-rich regions called the CpG island, which are typically 500 to 1,000 base pairs long. And more than half of gene promoters contain CpG island. And when CpG island within these regulatory regions, especially promoter regions, become methylated, transcription factor binding can be inhibited, leading to change in gene expression. And why we need to detect DNA methylation? The studies have shown that demethylation plays a very important role, and it can participate in maintaining the stability of genomic, and it also can participate in the cell type specification and genomic imprinting and the chromosome inactivation. And it also related to embryonic development. and the demethylation typically acts to replace gene transcription as gene splicing. And overall, the demethylation plays an important regulatory role in life, and it considered as a fifth basis. Next, let's take a look at the major technologies used to detect demethylation. So is WGBS comparing with other technologies here? Is including methylation macroarray, mid-deep-seq and targeted bisulfite sequence, and reduct representation bisulfite sequencing. And so WGBS is used to detect the methylated cytosine by treating the DNA with a bisulfite before we do the sequence. And it has a very high sensitivity, and it provides genome-wide coverage and can detect regions with low demethylation levels. And so the WGBS have become the golden standard for studying on genome-wide methylation at single base resolution. So in recent years, a large number of studies have used WGBS technology to detect demethylation. The main research directions for WGBS including such as cancer research, including early cancer detection and biomarker discovery, and growth and development, such as embryonic development and cell differentiation, and environmental impact, and also about the reproductive genetics. So in the next part, we will see some research cases in these different fields. And the first topic I want to talk is about the tumor early screening biomarker. Here we can see two pictures here. In first figure, we can see the major mechanisms involved in tumor development. Both genetic alterations and epigenetic changes can disrupt the normal gene expression and contribute to cancer. And moving to the next figure, we can see that our epigenome is continuously shaped by genetic and environmental and lifestyle factors throughout our life. And these epigenetic changes can be detected in accessible tissues and may serve as valuable biomarkers for cancer risk assessment and early detection. So there is a type of DNA called cfDNA in the body, which means the circulating-free DNA consists of fragment DNA released from cells into the bloodstream and other body fluids, and it has emerged as promising molecular biomarker for cancer detection. And circulating tumor DNA or ctDNA, which means the cfDNA is specifically derived from tumor cells. And because of its high stability and tissue specificity, cfDNA methylation have become an attractive biomarker for cancer screening and early detection. And during the early stage of tumor development, epigenetic alterations occur before obvious clinical symptoms appear. And these altered DNA fragments are released into the circulation, providing valuable molecular information for early cancer detection, diagnosis, and disease monitoring. And so about the non-invasive liquid biopsy technologies, targeting cfDNA have become one of the hotspots in the field of tumor research. And because of its advantages, cfDNA has been widely used in different fields, such as early screening and diagnosis, minimal residual disease monitoring, and the identification of the tissue origin of ctDNA. So next, let's take a look at some cases. The first is a paper about the researchers' teams had developed a multimodal method called the spot mass. And like the workflow shows here, it detects the tumor by looking at the methylation. It checks the methylation of the DNA copy number and the end part of cfDNA. Finally, the authors shows that the spot mass combined multimodal analysis of cfDNA features with innovative machine learning algorithms for early cancer screening. And another multimodal-based cfDNA has also demonstrated promising performance for early cancer detection and screening. First, they identified the ESCC-derived DMRs and the CMVs were from paired tumor and matched adjacent normal tissues using WGBS and WGS from 155 patients with ESCC. And finally, the biological significance of the optimal DMRs was investigated using paired ESCC tissue samples. And the next study, the authors collected from Chinese patients and randomly selected healthy controls, results in a training cohort and a validation cohort. First, they compared about 485,000 CPG sites between the liver cancer tissues and normal samples to identify the most differentially methylated sites. And then they picked out about 10 CPG sites to construct a diagnostic model and 8 CPG sites to develop a prognostic or a cellular production model. And because of the based on the diagnostic model worked very well in both training and the validation groups. And in addition, the model can also accurately reflect the stage of the liver cancer patient and also can tell us whether there is any residual tumor in the bodies and the response to the treatment and whether this is a recurrence or not. And in the next part, we will show the methylation signals can also be used for early screening and the diagnosis in the other disease. So first, we can compare the DNA methylation patterns in normal and aberrant DNA methylation cells, abnormal cells. And in normal cells, we can see CpG islands remain largely hypo or unmethylated, whereas gene bodies and another repetitive elements are highly methylated. And however, in aberrant cells, we can see the pattern becomes disrupted. We typically observe the global hypomethylation together with promoter CpGs and hypomethylation, which contributes to gene dysregulation and genomic instability. And next, let's look at a study about preeclampsia. The study included 64 women with PE and 38 healthy controls in the discovery cohort. And the researchers then analyzed the plasma cfDNA methylation signatures to identify methylation signatures associated with PE. And based on these methylation signatures, the researchers developed an early pregnancy risk prediction model and currently identified about 72% of early onset PE cases before the onset of clinical symptoms. And the researchers also found that many demethylation changes in PE patients were placenta-related, particularly in early onset PE, which was associated with abnormal plant central development. And after identifying different CFD methylation between patients with PE and healthy controls at diagnosis, the researchers asked whether this methylation change could be detected before clinical symptoms appeared. And they then evaluated whether these methylation signatures could predict the future onset of PE. The result shows that cfDNA methylation signatures around 12 weeks of gestation could identify women at high risk of developing PE. And interestingly, the methylation signatures detected during the first trimester differed from those observed at the time of PE diagnosis, suggesting dynamic epigenetic changes during disease progression. And finally, the researchers further characterized these differential methylation signatures by analyzing their tissue origins, associated biological pathways, and genomic distribution. And the researchers then evaluated the model in an independent cohort, nearly 200 pregnant women. The results were consistent with those from the discovery cohort, demonstrating good predictive performances across different clinical subgroups. And in addition, the cfDNA methylation score gradually decreased with gestational age in the control group, whereas it remained relatively high in patients with PE. And the researchers suggested that the model built complement exciting PE prediction models. They also proposed that incorporating demethylation signals could further improve PE risk prediction. And beyond the disease applications, demethylation also provides a powerful way to estimate biological age through epigenetic clock models. The first figure illustrates how epigenetic clock models are developed by integrating age-associated demethylation signatures with cell type and function-specific methylation patterns. The second figure demonstrates that these epigenetic clocks can accurately predict biological age and have broad applications in aging research and cancer studies and the evaluation of age-related biological processes. And in addition to being involved in disease occurrence and development and growth aging in the animals in the demethylation, it also plays an important role in the plant research. So in terms of plant growth and development, although the functions of the 5-DNA methyltransferase have been extensively studied, but their regulatory mechanisms are still not fully understood. So to solve these problems, the article uses a completely new method. The authors successfully construct a plant model linking demethylation. And by using the gene detection technologies, the researchers precisely targeted the methyltransferase genes to regulate plant growth and development without altering global methylation patterns. So this research material provides the scientists with an ideal model to detect the function of demethylation in the plant gene expression and the development fields. And the last two papers focus on the role of demethylation in environmental stress. Among different environmental stress, drought is one of the most severe factors affecting plant growth and crop productivity. So to cope with the drought stress, plants have evolved complex regulatory networks that coordinate stress responses. The first study summarized how demethylation participates in these regulatory networks during drought stress. And the second study further demonstrates that the change in the demethylation and mRNA or gene expression is associated and under drought and salinity stress, highlighting the important role of epigenetic regulation in plant stress adaptation. And so it's all about cases studies on the common research directions to the demethylation, and we classify the research content according to the omics technology. So here is two examples for multi-omics study of using epigenetics context and RNA-seq to explore epigenetic features. And through the demethylation analysis, we can identify DMRs, their associated genes, and the genomic features where they are located, such as promoters, enhancers, and other regulatory regions. And the transcriptome can observe whether the expression of genes has increased or decreased. So by combining these results, we can obtain and the expression level. Further analysis for the functions of these genes can help us better understand the biological mechanism behind the phenotypic phenomena. And in addition to combine with the transcriptome, the demethylation can also be combined with more epigenetic technologies, such as ChIP-seq and ATAC-seq, and also can include the HiSeq to achieve the research goals. And in the study, the researchers used a tool about multi-omics technology and found that the G9A shows the regulation of epigenetic modifications, transcription, and chromatin structures through the different mechanisms. And there is another example for integrated multi-omics analysis. So by integrated epigenomic and transcriptomic analysis, reward how intrinsic epigenetic regulation and the tumor immune microenvironment jointly derive subtype heterogeneity and safety response in pancreatic cancer. And next is the part is summarize the overall workflow for demethylation data mining, and it will generally follow the several steps. And the first step, we perform a genome-wide methylation profiling to obtain an overall view of the methylation. And the second part, we identify differential methylated regions and genes between groups. And finally, methylation data can be integrated with transcriptomic data to investigate the relationship between demethylation and gene expression. And the right side in this slide shows some representative examples of the analysis results. And let's go more to the final part, and there is a results validation part. The method of verification of demethylation includes a target gene demethylation sequence or TBS technology. And we can also use the qPCR technology to detect the mRNA expression level of the target genes. And we also can detect in the Western blot to make sure the protein expression. And there are also a more advanced validation method, including the knockdown, knockout, and overexpression of DNA methyltransferases. And also, we can do some DNA methylation inhibitors. And in addition, we can combine this analysis with experimental validation sequencing validation method and various sequencing platforms and are shown here. And I think that's all about the webinar I want to share with you. And thank you for very much for the listening and watching. I hope today's webinar sharing can be helpful for your research. Thank you.